Good afternoon, everyone. My name is Olivia Wilkins, and I have the pleasure of hosting the third installment of the Plant Cell Identity webinar series. Over the course of this series, which will continue for the next several months, we've been discussing what makes a plant cell identity. And so how can we distinguish between different cells, cell types, and different cell states? And so we've addressed questions through these various seminars along the lines of, can we identify or define a cell type based on where it is, based on which cells it has differentiated from, whether it has been conserved across evolution, whether we can define it by what it does or how it responds to different signals in the environment. And we have a lot of discussion. It's been amazing already to see sort of the diversity of thoughts that people have brought to this task. And so we are looking forward to today's speakers' contributions to this discussion as well. So before we get started with today's session, I wanted to highlight the next two sessions on March 11th and April 8th. So on March 11th, we will have Dr. Keiko Toriai and Dr. Justin Wally speaking, and they will be bringing questions of development and proteomics to the discussion of the definitions of cell identity. And on April 8th, we will have Dr. Jazz Dickinson and Dr. Nemanja Vukasonovic, who will be looking at live imaging revealing new cell types in the Arabidopsis root meristem. In addition to the Plant Cell Identity series, we have the Plant Cell Atlas Spatial Metabolomics Workshop is coming up on May 9th. And so this is a virtual workshop and everyone is invited to join this. You must register in advance for this workshop. And so I invite you to go to the Plant Cell Atlas events page where all of the registration information is available. And to please register. I think it will be a really spectacular series and sort of bring a new dimension to the way many of us are thinking about space in plants. So with that, I would like to begin by introducing our first speaker today, who is Dr. Kenneth Birnbaum, who is a professor at the Center for Genomics and Systems Biology at New York University in New York, and he is presently the interim director of the center. Ken received his PhD from NYU, and he has produced really, over his career, a really spectacular array of resources and sort of innovations in thinking about plant development and tissue regeneration. He is also a member of the steering committee of the Research Coordination Network for the Plant Cell Atlas, and so has really been fundamental in sort of building the framework through which today's series is a small part of the Plant Cell Atlas work. And so with that, I would like to hand the floor to Dr. Birnbaum, and I will then, I should also say, there is a second speaker, and the way we will be organizing our session today is we'll have Dr. Kenneth Birnbaum's talk, followed by a talk by Dr. Kaisa Kajala, and then we will have questions at the end of both talks. Great, thank you very much. Okay, I'll stop sharing.
Okay, thanks everybody for joining. I'm going to address this question of cell types versus cell states based on a lot of data we've collected over the years and sort of thinking in this area. And I would mostly like to instigate a healthy discussion on this because I think these questions are still very much open. Okay, so what's the difference between cell type and cell state? I have the happy face up there. This is a question that I love to ask because it really brings out a lot of ideas. But then it's a question that I really leads to some consternation to answer because there aren't easy answers, and I'll go through a little bit of what I mean by that. I'm going to follow Natanella's prompts that she gave me because I really like these questions of how I'm defining cell states and cell types now. How could we better define them in the future? And why is it important? Which I think is a really good question. So let me start in by just saying we are not alone in asking this question. Many of these issues have come up, and of course, all related to the technologies that we have here on the plant side. Adrian has really probably stated it very well by, well, it's complicated. We've gotten some interesting points of view from Jeff Doyle and Mark Lebeau, comparing this in a sense or as a metaphor to the question of evolving a new species, of how you classify or think of a plant cell in that context. And then the animal people have contributed to this in various ways. Lineage, of course, is a big deal for them, and function, and this concept of a core function will come up, and I will talk about that too. Anyway, okay, so a lot of why we raise this question has to do with this series of new technologies, sets of new technologies, and single cell RNA-seq notwithstanding, not the only, but one of the major ones, and really interesting perspective, right, where we can group these cells and we can actually sort of now visualize the difference between them, and we have this tendency to, want to say that if there's a distinct cluster here, that's a distinct identity. And perhaps, naturally, if we see clusters that are closely related here, these cortex clusters in maize, we might want to think of these as subtypes or states of the same cell type, right? So that's our new perspective, and I think it's really powerful. But at the same time, I think we have to ask: is this the right data to ask that question? And then is then distinguishing between cell types and cell states actually important or useful at all? But I would argue that it still is. And even if we cannot answer it, it actually leads us in a good direction, right? Because we're asking about cell function, and we're asking about the distinction in cell functions. And in the process of trying to answer that question, I think we approach many interesting things about the cell and differentiation and evolution and all that. And I think, yeah, this might not be the right data, but it's kind of the best data we have right now. And so acknowledging that this may not be the last word in answering this question, I think we should use it to our best ability and move on, and allow new technologies to supersede our conclusions later. Okay, so first, I'm going to just make the intuitive case, which I think everybody can relate to, for cell states versus distinct identities based on mainly work that we've done over the years now. And so, for example, this is old data going back to the days of cell sorting and cell sorting specific markers. And for about 15 years, right, we were using many of the same lines, especially in the root, to sort cells under different conditions. So we have comparable sets of cells, not individual cells, treated in different conditions. And so this is one of the concepts that, right, we all kind of can believe. Here we see in the cortex marker, the controls all clustering here, for example, and here cells, the root treated with iron, and then we are actually Jose Dineni profiled these cells, and then I'm sorry, that wasn't Jose, but anyway, Jose was with these, did the salt, and so we see, right, the same cell type moves a little bit. It doesn't become another cell type, but it shifts its position in this hypothetical principal component space to a new place. And so this is, epitomizes the concept of an environmental state where yes, you respond to your environment, but you don't necessarily change your identity. Here's some work, or sorry, this is a view and sort of inspired some work that we did where, of course, the maize root and many monocot root and many other plants make different kinds of roots, right? Primary, crown, seminal root, laterals. And they have a very similar cell type, cell anatomy, cortex, endodermis, et cetera. But they have different functions, right? And the overall anatomy can differ somewhat. Okay, so Sunil Kenchanmani Raju in the lab when he was there decided to take a look at these and look at the difference in the single cell composition and expression of these cells. And the first thing that you can see here is that he's colored in different colors the samples of single cells that came from the different roots. And what you'll notice here in the integration is that these pretty easily integrate together where, you know, the cells in this particular cluster come from all the different kinds of roots, meaning that it's the same cell type, whether it comes from crown, primary, seminal, or lateral roots. And maybe that's not so surprising. That is same cell type for the sake of integration and clustering. Maybe not so surprising. But then what Sunil did and what can be done, right, is to take an individual cluster and now you can subcluster that. And you can see that the cell types start to separate, right? The crown root cell types are here for the endodermis. The main and primary roots are, sorry, the primary, sorry, this is, yeah, mains, and the laterals are here for the endodermis. Okay, so one might call these subtypes or maybe developmental cell states in this case. And then, you know, the final example in Bruno Guillotin's paper here, we took maize, sorghum, and Soteria. These are related species, right? There's only about 60 million years here, but we could map the orthologs well, and so make this comparison, and we can combine it. And indeed, again, the cell type of the individual species will cluster together. And so one would want to think, as anatomy has told us for many years, that these are homologous cell types, you know, with somewhat different expression that represent the same cell type, but perhaps different evolutionary states. Okay, so that all to say is I have an intuitive sense, and I want to believe that there is a distinction, that there are cell states and not only different cell types, and that we can somehow distinguish them. Okay, so if you want to go down that road, it seems like the first important thing to do is come up with some quantitative way, because now we can do it, right? We have all these quantitative outputs of cell identity in many, many dimensions, right? And so we should come up with a measure. We should be able to come up with a measure to do that. Okay, so this is an approach that we developed, or actually Jesse Gillis developed with Michael Pasalacqua in his lab. And it was called, or is called, the meta-neighbor approach. And it was originally designed to ask whether clusters in two different data sets really are equivalent, and to come up with a quantitative way to ask about that difference, rather than just try to integrate everything and force everything together, which maybe you could do, but you're losing information, right? So the method, which I'm going to really shorthand here, it's much more complicated than what I'm saying, and has a more elegant formulation. But for example, if you could map, these are two data sets, and there's different ways to map the same genes in another data set, either by homology or maybe they're the same genes. And so you can ask, well, is any given cell in this cluster map most closely, is its neighborhood, meta neighborhood, the cells of another given cluster. And you may define this cluster ahead of time, or you don't have to. You can discover it a priori. And so that's a nice match, right? And you could go cell by cell and do the same thing. And then let's say there's another cluster or another outcome where it's a little sloppier, and this cell doesn't always map to the same cell in this cluster, the same cells. And the strength of this may be this might be its closest match. So then, that's a poorer score, right? And then that's an even worse score. So you can quantify all this. The way Jesse does it is actually sort of build this network and look at the connections of individual cells to other clusters, and that gets taken into account. But one could easily start to do this with different species and start to be able, for example, on an evolutionary level, map the distance between cell types. And we did that. And here you can see. So what we did was ask from the perspective of Soteria, what's the difference in individual cell types with respect to maize and sorghum, right? And so now we can quantitatively ask that question. So for example, epidermis is quite related, right? There's not a lot of so-called distance between the cell types in terms of their transcriptional identity between these species. Steel seems to evolve, if you would call it that, a little faster, and columella, for example, seems to evolve the fastest. And then there's a difference between the, let's say, distance between maize and Soteria than there is between sorghum and Soteria. So one might want to say here that, perhaps the maize is evolving, the maize columella is evolving faster. I know there are a lot of caveats to that, but this is the kind of view that a quantitative assessment of cell identity can get you. And then you can dig in there, and here we used Mini-X to ask about transcription factors and their potential targets or their predicted targets, and make modules and ask about what these modules of gene expression are doing as these cells change, right? And so this is just an example. Transcription factor may have a set of genes in cell type A, and you can track that transcription factor and all its targets. And Mini-X may tell you, yes, I see that transcription factor and all its targets, so that module is there. And then perhaps in a second species, that transcription factor and its module, its downstream genes, are now in a different cell type. So we can do that in mass, or for all the genes, or all the modules that we can actually track across the species. And so, for example, here you see that what is going on is whatever module this particular transcription factor controls. You see it in the phloem in all the three different species, sorghum, Soteria here. But what you see quite often are things like this, where you have a module in the cortex in Soteria that has moved, or is expressed somewhere else in maize, and that somewhere else in this case happens to be epidermis. Or here, where in sorghum there's a module that's expressed in the phloem, it's now in or it is in Soteria's pericycle. And this switching around actually turned out to be, or the swapping as we called it, turned out to be much more prevalent than the conservation. And digging into this, one of those modules was a mucilage expression module, that is a mucilage genes that are involved in mucilage. And here's a heat map of their expression, and here are all the cortical cells. Here's the columella cells, the columella cluster. And you can see in Soteria that most of the mucilage is expressed here in the cortical layers. I don't know why not one layer, but among those layers. And then in sorghum, they start to move into the cortex here, but then in maize, they're virtually solely in the in the columella. So there's some kind of switch, there's some kind of swapping between. Here we can sort of put our finger on a function and track it and see that modules change. And this very much coincides with what we know about maize, of its very sliminess in the cortex. Here's one landrace that really does that a lot. And so, it seems to have swapped from, or this component of cell identity seems to have moved from the cortex to the columella, or, well, it seems to be the ancestral state in the cortex. Okay, so all this by the way of saying, and I actually believe this because I think this is something that I really first, well, uh, I feel like this theme comes up over and over again in the research that I've done over really like the last 20 years, is that there's a lot of aspects of cell identity that fall into modules and seem to get swapped around. And so I feel like maybe one way to contribute to this concept of this definition is that gene modules are the currency in which cells change. Of course, genes are important, but maybe we can go a level up and look at this modular level. And so my analogy here is the Lego view of cell identity. And I think this was actually the Lego view in one of my first presentations when we started all the cell sorting stuff. And you can start with identical cells as these are supposed to be, and then you can slowly change them, right? A cell doesn't have a single identity in a sense, or a single functional genetic module. And so over time, that may happen, right? And these modules seem to pile on. So that raises the key question of when is the module, I've kicked the can down the road here, right? When is the module composition different enough to call a cell a new cell type? And the answer to that is, I don't know. But it's something we can start to ask, right? And if we take it from the point of view of modules, we can do it. Okay. And then, other caveats. Well, can you stay in the same anatomical position and diverge in function? Here, I'll bring up some of Kaisa's old work, really elegant work of looking, this was still in the cell sorting days, but looking at the different cell types across tomato, Arabidopsis, and rice. And, a lesson that came out to me, and which they stated very clearly in the paper, is that, you can have the same cell type and the composition. I mean, this is a very broad evolutionary distance, right? And we've noticed this too, is the composition of the cell types. Presumably, a lot of the function should remain the same. Here are cortex cells, and yet they don't really look anything like each other across the species. Of course, there are all kinds of mapping orthologs or homologous genes, but this becomes really obvious that the transcriptional identity of cells is really hard to track as you move across evolutionary distance. And I think this is a good warning to us that probably that first point of, is our single cell perspective at the transcriptomic, even the proteomic, or maybe the epigenomic level, is it really sufficient to tell us if a cell is still the same cell identity? Because the contents we're measuring could be very different. But with that caveat, here's kind of a similar thing that we did in Carlos's paper, where we looked really hard at markers in Arabidopsis and tried to track the ones that were indeed conserved in the maize. You see markers, for example, cap markers that seem conserved across this huge evolutionary distance. So I think one of the things that we can do with that question is flip it around and say, well, if we assume perhaps by anatomical position and other evidence that these cells do have a similar function, can we define core functions by comparing them and looking at their common modular components? And perhaps there are genes that signal that core function. And maybe those genes will help us define what a cell identity is. Okay, so just quickly here in the last couple of minutes, I don't want to take up too much time. I'm going to go very quickly here. This is Sunil's work, which we have not yet published, but we're now looking for conservation across a long evolutionary distance of the shoot and the root, and we can look for genes that have conserved correlation environments across those different organs in a cell type by cell type manner. Okay, one of the things that comes out here is grassy tillers, well-known domestication gene. Interestingly, we see differences between the seminal and the primary root and the lateral. Here it is in the, I see my internet's unstable. I hope you can still hear me. I'm going to keep going. And so here you see in the cell types that are maturing more quickly, very interestingly, it expressed in the primary. In the lateral here, you see it spread through many, many cell types. And so this leads to the conjecture that perhaps what grassy tillers is doing is, which it does in the shoot, which is sort of make the cells more mature, maybe prevent as much division is happening. And long story short, yes, if you look at the grassy tillers mutant, you see the laterals now growing out more. And so these cells have been told, or now don't have the instruction to halt the cell cycle. And so they're growing out longer. Okay, so the main point here is this we can believe is the same cell type. Ground tissue in the primary is the ground tissue in the lateral. But here you have a gene that seems to be commonly used in evolution to tweak the state of the cell. And so maybe there are genes I would throw out there that are cell state modulators, switches, toggle switches. They don't need to just add on new functions, but maybe affect the core functions of the cell. Okay, and now just last slide here. In this attempt of making a definition, or what is my definition, as Natanella asked me, maybe here's one where I would call a cell type. And the stress here is that we should put the definition on the functional level. And then letting our data just try to inform that rather than having our data define the distinction. Okay, so any group of cells that share a core set of functions where, yes, anatomy and position matter, but not necessarily dictating the distinct roles because, well, cells can change a lot in the same position. And cell states, different chemical states, well, some, transcriptional, protein, that respond to, of course, environment, organ type, evolution, where there's still some core identity. What is that core identity? Yes, I'm going to have trouble defining that. So I think these are ideas that have come along already. I just want to stress that I would want to keep these at the functional level. But here I would, in the sense of improving, how can we improve this definition? Maybe we can just really start our understanding of what all the genes mean, and that is understand the function of these gene modules better, because that seems to be one of the major currencies that the cells are using to diverge from each other. And in that sense, what would be needed is more high throughput phenotyping of cellular functions. What do these modules do in cells? And so then we can interpret better when we see differences. Is this indicative of a cell state change or a cell identity change? Okay, I'm going to stop there. And I'd love to have questions, have people, dispute any of this or all of it. And thank you. I think I recognized a lot of the people for the work during the way. So I'll skip the acknowledgements and I'll hand it over, and really curious to hear Kaisa's presentation. Thank you.
Great. Thanks so much, Ken. That is tons to think about. And I think there will be lots of questions and discussion for you. So this is just a reminder to folks to please use the Q&A if you have questions, and we will get to them at the end of this discussion. But also in the chat, there is a link to a Padlet. So for those of you who have been at past sessions, you know how productive that discussion can be. And so basically, it's a forum where you can start posting questions and answers to one another in real time. So it's a great way to have lots of side discussions on the go. And so again, that link is in the chat. So it is now my great pleasure to introduce Dr. Kaisa Kajala, who is an associate professor in the Department of Biology at Utrecht University in the Netherlands. She received her PhD from the Department of Plant Science at the University of Cambridge. And her research is really dedicated to understanding how different cell types have evolved. And within that, she also is looking at how these cell types develop in different plant types, how they respond to the environment, and how changes in these cell types and their sort of distribution in tissues contribute to plant fitness. In addition to sort of her fantastic scientific work, Kaisa is a really dedicated mentor and has put lots of effort into building strong interdisciplinary teams to support research and training. And so it is my great pleasure to welcome Dr. Kajala to speak next.
Thank you so much for the really kind introduction, and thank you for giving me the opportunity to talk about my opinion about the plant cell types. I trust you guys can see my screen. Yes, awesome. Okay, so today I'll be also following some of the prompt questions that were given to us ahead of time, and I'm going to start by the question, why is it important to define cell types? And for me, it's really important because it's the basic units of interest in my research program. So we study root cell types, and the key questions for quite a while now have been, how do cell types know who they are? How do they integrate environmental cues into their development? And how have these processes evolved? And specifically, we use the barrier cell type Exodermis as our model cell type for understanding these things. So given that we're interested in evolutionary processes, we need to be able to define cell types in a way that holds between species. And this is the difficult part. So it's very easy to define cell types for single species. But as soon as you have this comparative aspect, I find that it becomes really, really difficult. So some of the traits that I think can be used to define a cell type in some cases, and some cases not. So in the case of Exodermis, so firstly, position. Where do you find this cell type? So Exodermis is defined to be the outermost root cortical layer of some species, not all. So position here is not sufficient as the definition. You can use morphology. So differentiated Exodermis accumulates lignin and or suberin in its cell wall. Well, so does endodermis. So the position here with morphology works to define these two cell types. But also there has to be a function that Exodermis and endodermis, they function as apoplastic diffusion barriers to water, solutes, and gases. So in the case of Exodermis, like with these three traits or aspects, it actually works really nicely. But as a counterexample, I'll introduce you bundle sheet cells. So position surrounds, bundle sheets surround the vascular bundle in the leaf. That's pretty strong. But then if you look at the morphology and function, these are really varied between the leaves you look at, based on if you look at a C3 leaf or a C4 leaf. So in the C3 leaf, the bundle sheet cells are fairly small and have roles in transport and metabolic processes involving sulfur and nitrogen. Then in C4 leaf, like you can see here on the right, you have the bundle sheet cells that have evolved to become powerhouse of photosynthesis. So C4 leaves have an additional biochemical cycle that concentrates carbon dioxide into the bundle sheath. So you see the enlargement of the cell, and as well as increased numbers of chloroplasts here clustered around the vein here, so the darker dots in the cell. And in some lineages of C4, you also see tuberization. So you have this like really diverse morphology and function, but everyone always talks about them as bundle sheet cells. So in this case, the morphology should take into account all of these diverse morphologies and diverse functions of the cell type. So here I think that it's okay to talk about the accumulated functions, and then what becomes, in addition to the position, something that could be used to define the cell type is the homology. So they are of shared origin. So it is the same cell type that has just accumulated additional processes. However, homology, I'm not a massive fan of, because there are parallel evolutions of things, such as roots. So if we start saying that, let's say a root cap cell, to be a root cap cell, it has to be from a certain evolution of roots, then it makes it really difficult to specify cell types without knowing their evolutionary past. So yet again, definition that may be useful or may not be useful. Then let's go to the transcriptional aspects of the cells. So marker genes have been traditionally being used to describe cell types, and now with the single cell sequencing, the transcriptional phenotype. So again, actually really nicely displayed many great examples of this. And as the counter example, I didn't actually even realize that I could have brought my own example. I actually brought something else. So this is a recent preprint that I really liked, where they carried out single cell sequencing of 4 different Brassicaceae species, that some of them are extremophiles. And here's a really cool example of how the cellular transcriptomes cluster within the species. So the transcriptomes of plants actually evolve really quickly compared to, for example, like if you compare flowering plants and mammals. So plants have way faster evolution of the transcriptome, which leads to the fact that their cellular transcriptomes, tissue transcriptomes, end up resembling of the transcriptomes of the same species more than the corresponding cell type or tissue type from the other species. So you can't exactly use this easily to specify cell types. Okay, so then these sort of transcriptional phenotype or the marker genes, they come from regulation, like what regulatory complexes or networks are present in a specific cell type. So to be a cell type, though, I think it's important to have regulatory distinctiveness. So that cell type has their own regulatory program that is different from the other cell types. And in the animal field, and especially when talking about the evolution of animal cell types, they have discussed at length the idea of core regulatory complex, which was defined as the physically interacting transcription factor complex that enables and maintains the distinct gene expression program in a cell. And then later on was refined as the core regulatory circuitry, a set of transcription factors and interacting factors that together define the gene expression profile of the cell, and which is the framework for defining cellular identity. And I don't actually know how well this has been studied to the level that we could actually name any of these conclusively for a cell type. I think that the short root scarecrow module could be used to define, to some extent, fulfilling the role of this, and that seems to be conserved quite widely in the angiosperms. So it does meet some of these requirements, but still, I'm not sure how conserved they are across like evolution when it comes to comparing, for example, let's say Exodermis in one lineage to Exodermis to another angiosperm lineage. So what can we do with this lack of definition of cell type? So we could accept that biology is not always categorizable. and cell types are fluid. There is beauty in biology and its fluidness. And even though the cell types evade our willingness to categorize them, we can still ask really exciting questions about their biology. So we can still study how cell types evolve, but I think that we just need to be careful to break the questions down to specific processes we are interested in. So we can still study, for example, a specific regulatory cue that specifies a cell type, and we could follow the evolution of that cue. We can look at cell type specific gene regulatory networks and how the wiring of those networks have evolved. So I think that needing to specify a cell type, it might be the problem. So I don't think that they're like, as Ken said, that we don't like, he doesn't know how to really specify things or define things that like carefully. I'm having the same problem, and maybe I'm making the argument that maybe we don't need to necessarily. But as a case study of how to study evolution of cell types, I'd like to present Exodermis. And these pictures, actually, the suberin and lignin stained images are from chickpea. And I currently have a project running looking at the Exodermis across flowering plants. So with my wonderful team of Leonardo Joe and Brianna Kluk, we've been looking at few different angiosperm lineages or families, specifically grasses, cabbages, and legumes, which have been previously recorded to have both exodermal and non-exodermal species. And in contrast to the previous work, we are actually looking at these roots not only in control conditions or happy conditions, we also are hoping to stress the plants a little bit with fully artificial ABA kick to see if they can deposit the exodermal barriers in response to stress. So ABA is known to regulate suberization of endodermis in Arabidopsis, but also the exodermal and endodermal barriers in variety of species. So we don't want to miss out on any sort of potential, because it's difficult to see the cell type unless the barriers are actually present. So I'm going to today actually focus on the legume story, and we have a little preprint out of this as of January. So a lot of the data that I'll be showing can be found from the preprints as well. So this is how we screen our plants. So as an example, a chickpea, so we grow them for a few days on plates, then give them a 24-hour treatment of either control, so half MS, or 2 micromolar ABA. And then we section and stain the roots, looking for lignin stained by basic fuchsin, or suberin stained by floral yellow. And then we quantify the intensity of the signal. So for example, here we see ABA responsive lignification and suberization in the outermost cortical layer. And this is what I expected the project to be like, very clear, barrier, no barrier. But what it turns out that, yeah, the cell types and the phenotypes are pretty fluid. So it was quite difficult to call which lineages actually had outermost cortex carrying suberin or not. So here is the chickpea again. But then if we look at these other species, we see in P the same phenotype, exodermal suberin. Then in Viscia sativa, we see induction of endodermal suberin in response to ABA, but also induction of signal picked up by floral yellow in multiple layers, including the epidermis and 3 of the cortex layers. In Medicago, which is strongly known as endodermal species, we also picked up some very weak suberin signal or floral yellow signal in the cortical layers. This is, by the way, something that we would have never picked by eye, but the image analysis gave us weak level of signal. So once again, we wanted to be like, let's do statistics so we can be conclusive. And it turns out that the statistics pick up things that we would have never called by eye. So we basically have 2 exodermal species identified, chickpea and pea. One where we saw strong multi-layer formation, and then a number of species that we think are endodermal. And then we followed up 3 of these species, chickpea, Vishya sativa, and medicago, as our sort of triad of exodermal, intermediate, and non-exodermal species. We wanted to confirm that the suberization really is real, because histochemistry has its limitations, and sometimes they indeed pick up other compounds than the intended one. But with the transmission electron microscopy, we picked up suberin lamella in the ABA-treated exodermis of the chickpea, here seen in these beautiful wavy ripples. In medicago, where we were not expecting to see anything in the exodermis, we did not see suberin lamella. And in Vishya sativa, the one that had the multi-layer signal, we picked up something that could be suberin, but it definitely was not suberin lamella. So we are thinking that maybe there are some suberin monomers being deposited, but no lamella formation. So we don't know if it's sort of suberization pathway gone wrong, or sort of building up the pathway to the point where it could form a lamella later on. There's multiple hypotheses in the lab about this at the moment. Then going to, so that was the phenotype, but then going to the function. So we also wanted to test if the exodermis in this species works as a functional barrier. So we carried out a penetration assay where we tested if the exodermis can block the diffusion of propidium iodide. So we treated entire seedlings, or we basically put entire seedlings in PI stain for a short period, and then rinse them many times to get rid of the excess dye. And then we sectioned and imaged where the dye had penetrated. So in medicago truncatula, we see that from epidermis through all the cortex layers in both MOC and ABA, we see the stain marking all of the cell walls as expected. So no blockage of the dye penetration. In chickpea, we see that the dye is blocked at the outermost cortical layers, and it's slowed down, and no PI signal is picked up in the inner cortex layers. So here we have a functional barrier. And then the interesting one, Vishya sativa, here we actually do not see any blockage to the PI penetration. So whatever the suberin or suberin-like compound is doing, it's not functioning as a apoplastic diffusion barrier against PI. So based on this, I would call only the chickpea having an exodermis, and I have no idea what Vishya sativa has. Okay, so then we want to move, we had a model basically, so there is an ability in chickpea to differentiate the outermost cortical layer into an exodermis with a barrier function, but of course the entire root has general ABA root responses. So to distinguish between these 2 ABA response programs, we set up 2 different transcriptomics experiments for these 3 species to infer their gene regulatory networks, and then to be hopefully able to compare how these have evolved for the exodermis function. So first experiment was the time course, second one was a single cell, and at the moment we have just received the data for Vishya sativa and medicago truncatula here. So today I'm actually only showing the chickpea data. So I realized that I'm running a little low on time, so I'll be quite quick about this. So I'll show that we found in our time course data basically 4 separate clusters of upregulation: fast, medium, and slow. And then the general ABA response is in the fast cluster. The suberin biosynthesis induction is in the clusters 2, 3, and 4. And then what regulates these based on the motifs enriched in the promoters. So the cluster 1 had enrichment of the G-box motif, so the ABA response elements. So most likely immediately downstream of the canonical ABA signaling pathway, whereas clusters 2 and 3 had an enrichment of the MIB motif, and MIBs are known regulators of suberin pathway in multiple different tissues and species. And what we also see is that there's a slew of MIBs that are regulated at different time points, including 2 who are regulated very early on at the 45-minute time point. And one of these had 2 of their G-box motifs in their promoter. So it's now our likely candidate. Okay, so then
Kaisa, I'm afraid your slides have dropped out. Are you able to put them back up, please? Thank you.
Yes, I am. I don't know what happened. It suddenly said that I'm being recorded. Share screen, where is it? Here. Choose a screen. No, just a second. Sorry, being a little slow. Yes?
Back up, yeah. Thank you.
Yeah, sorry about that. Okay, so then we carried out a single cell sequencing experiment to see how these different suberin and MIB genes are expressed spatially and if they are in the right location. So we generated a chickpea root atlas in the 24-hour ABA treatments. And we were able to annotate a lot of these clusters based on pre-existing Arabidopsis markers. So having said that, maybe transcriptome phenotypes are not the most reliable things. Regardless of that, it was very easy to rely on the body of knowledge from Arabidopsis, and it looked very straightforward for many of these. When it came to the barrier cell types, we had to add the fact that because Arabidopsis doesn't have an exodermis, so we looked at what was the endodermal, exodermal traits in chickpea. So in chickpea, we have a very specific Casparian strip, whereas the lignin in the exodermis is not positioned the same way. So we were able to use the Casparian strip related genes to annotate the endodermis. And then as the chickpea endodermis does not suberize, we use the suberin to annotate the exodermis. And then when we looked at our ABA upregulated MIBs, we found that some of them were in multiple cell types, but some are specifically in exodermis. So to resolve the hierarchy of suberin regulating MIBs, now we have the time data, the cell type specificity data, and we have some of the trajectory predictions. So we have identified a trajectory where the suberin genes are starting to be expressed along the trajectory, which would correspond to differentiating exodermis. And for the MIB genes, early MIBs, early MIB, the 00232 goes also up early, followed by the other MIBs. So now we have a model where when all of this is put together is that we have this early MIB in multiple cell types, potentially due to the fact that we give a strong ABA kick from the exogenously from the outside. So maybe this drives it also in the epidermis, and in the epidermis we see bit of that network presence, but the full network only comes in the exodermis. And we think that we've identified the MIB that ties the ABA signaling into the suberin biosynthesis network or the differentiation network of exodermis, but we don't know what limits this gene regulatory network to exodermis only. So there is the spatial aspect that is still missing in our signaling network. So that's something that we hope to resolve in the future. But also at the moment, the work that we are carrying out is doing the evolutionary comparison between chickpea and these 2 other species. I'm going to skip over some of the tools that we have for testing the interactions in planta. I would like to thank my team, especially Leo and Rihanna, who've done a lot of this work. I want to highlight Leo's R package GG plum map that allows you to lots of wonderful heat maps in a shape of a plant. And then I'd love to take questions. No, we have a little bit of discussion with Ken first, I guess.
Yeah. Oh, thank you so much. That was an absolutely wonderful talk. There are lots of questions coming up in the Q&A, and I have lots of questions. So I'll save mine for another forum probably. But one thing that I was hoping that you and Ken would be able to address, I think that you sort of, you weren't at extreme poles in this, but I think that you sort of fell at different points on a line here, thinking about how important it actually is to define cell types in plants for being able to do the types of works that you think about. And so, yeah, I was just wondering if you wanted to respond to the position that each other
Ken,
had.
are you there? No, we've lost Ken.
Oh,
No, sorry. Go ahead. You guys, you go first.
No, I already claimed that we don't necessarily need to define cell types. We just need to define our questions in a way that, lets us not necessarily call cell types by any specific name.
Okay. Yeah. I mean, I'm not unsympathetic to that. I think that's valid. I just think that intuitively, right, as I made the case, it's probably good to think about it in the sense that we do want to know what core functions are, and we do want to know how much the environment is changing a cell. I mean, and that will help the line.
I fully agree with that. I just think that having to put a label on a cell type is really hard.
No, and I agree. And then where we draw that line, it's subjective. It's always going to be subjective, no matter what kind of data we have.
Yeah. No, sorry, go ahead, Kaisa.
I really agreed with everything with Ken said, presented in his talk. So.
And I agreed with everything Kaisa said. Even though he came
perfect agreement, but landing in different spots. I think you both brought really interesting perspectives. So just because we are so close to the time, I wanted to let all of the participants know that any of the questions that get posted in the Q&A, we will move over to the Padlet. And so Ken and Kaisa can answer the questions there. So your questions won't go unanswered. They just might not be answered live right now. But I have a bunch of questions, but I feel like I'll stick to the ones from the Q&A for these last couple of minutes. So the first question, I think without the figure might be a bit difficult to answer specifically, but when you have a cell type that has different groups, so for example, epidermis 1, 2, and 3, is it because they are sort of different cell types that we would define like guard cells, trichomes, and pavement cells, or is there some other reason why you might end up with multiple clusters of cells that all get called epidermis, for example?
Yeah, maybe that was in my heat map. I think that's a good question in the sense of how do we interpret our dot plots there, but I think very often they're developmentally closely related cells like cortex and all the different layers of cortex. But I think we have to be careful because sometimes they may represent different developmental stages, and then do we really want to call that a different subtype or cell type? Yeah, I think those are my two answers to that question. Kaisa, what do you have any input there?
I actually have to say that I would also expect the same, that it's either developmental or then when you have multiple cortex layers, I do think that they might have slightly different functions and identities and capacities. So I would expect to see multiple cortex layers in a cereal, for instance, or in a legume that has up to 15 cortex layers.
So sort of, I think a nice follow-on question from that, are cell-specific markers the same across different species?
Some are, some aren't. And depends how far you look.
Okay.
Okay.
Yeah. I like this question, and there was another one in the chat that asked me, I think, are some modules better than others at signifying identity? Like, should we value this particular module when we say, okay, it's just the same cell type responding to environment? And I think that's actually a really interesting way. I think it's a related question. And maybe, I don't know, I'll throw this also back to Kaisa. Maybe we can use evolutionary comparisons to ask what are the best conserve, right? Conservation is the key to function, or however we say that. And then we can compare that with environmental perturbations and see how those have changed or not changed. And so I think that's a nice question because it leads us to another way of analyzing our data in the sense of analyzing both evolutionary and developmental and environmental perturbation,
Yeah.
putting all that information together. Go ahead, Case.
Yeah, I agree. I like that. Like any way of like perturbing the system is always like good for finding out what's important and what's not, I think. Like any additional dimensions are always informative. And the evolutionary direction is always tricky.
No, excellent. So yeah, no, I'm trying to coordinate these questions thematically because I think that there are a lot of interest in sort of what really is defining the cell, unsurprisingly, given the topic of our meeting today. So another question that comes up is when comparing cell types between species using a subset of genes, for example, one-to-one orthologs, have you assessed whether this subset equally preserves cell type identity structure of the clusters within each species? They are wondering if the level of difference or similarities observed across species could be influenced by gene selection rather than reflecting true biological divergence.
That's a great question. So I have not recently done a lot of like one-to-one ortholog comparisons myself, but I have explored other people's data when I've been looking at the superhuman regulators, for instance, the MIBs, so how they behave in different data sets. And from many one-to-one ortholog sets, they are missing because they evolved in so many branchy ways that they, are just not in that data. So one of my pet peeves is one-to-one ortholog sets because a lot of the interesting stuff tends to get lost in those. So I would expect that there is a strong effect of what data you use and what connections you make between the evolutionary history of the genes for your comparison.
Oh, great. Ken, did you want to weigh on that?
No, I mean, the only thing I would add, yeah, I mean, I think the one-to-one is tricky, agreed. I think we probably I think this points to our need to really map functional orthologs a little better across species, and especially where we have all this duplication. And, something Sunil and I have been thinking about is this ortholog or homolog switching as you evolve. And there seems to be plenty of evidence for that. So, yeah, I mean, I think that raises that important issue of how we map genes from species to species.
Great. Thank you so much. And as I say, there are lots of questions that will be waiting for you in the Padlet if you have time to address them afterwards. But thank you so much, both Ken and Kaisa. Those were wonderful and really stimulating talks. I'm afraid we're going to have to draw the discussion to a close, though, right now. But I'd like to remind people of two things. Number one, the next set of talks in this series will be on March 9th. And then, of course, there's also the spatial metabolomics workshop coming up in May. And so you can sign up for that through the PCA events website. Thanks, everyone, for participating. Wonderful session today. Great. Thanks.
Thank you. Bye.